Okay, so my project was studying vinculin load-dependent alpha-actinin localizations and focal adhesions at the Duke University Cell and Molecular Mechanobiology Lab under the PI Dr. Brenton Hoffman, and my graduate student that I worked with was Arnold Tao. So there are 20 deaths per day according to the American Transplant Foundation as a result of a lack of organ transplant from donors. This opens up a big market for artificial organs to replace the lack of donor transplants. And the primary source of artificial organs would come from stem cells. And so researchers have found, interestingly, that you can differentiate stem cells and make them into whatever kind of cell that you want based on the stiffness of the material that you put them on. So if you put them onto something that resembles the stiffness of a muscle cell, then the stem cell will become a muscle cell. And likewise goes for something that resembles the stiffness of a brain, then they'll become neurons. And again, the same thing happens with something that is as hard as a bone, then the stem cells will become bone cells. But even more interestingly, the shape that the cell is patterned into also affects what they differentiate into. So if you have a cell that's shaped like a star, then they'll also become bone cells. And if you have a cell that's shaped like a flower, then they will become lipid cells. And this is all very interesting because it shows that cells can sense what type of material they're placed on, but we still don't know how exactly they do that. So what I did was study the focal adhesions. So this is a model of a focal adhesion that's used in our field, and the key points of this model are the extracellular matrix at the bottom, shown in blue here. That is what I've been talking about as the material that you place them on. It's kind of like a scaffolding within our bodies, and they have varying stiffness depending on what material it is. So that is outside of all the cells and within all our bodies. The next part are these red integrins here. Those connect through the plasma membrane from inside the cell to outside and act as a connection between the cytoskeleton of the cell and the extracellular matrix. Next, you have all these colored proteins within the cell, and the key thing to note here is that they're formed in layers, and you can see that they're linked together like a string, and that's because they're all force sensitive. So if you pull on them, then that's how they sense the outside of the environment, and then they can test the elasticity of the extracellular matrix. And the most important one to note in here is vinculin, shown in green, because those have been shown to be a key protein within focal adhesions that act in focal adhesion assembly and disassembly, and a variety of other factors. So we already know that it's key to force transmission within the cell because vinculin is force sensitive, which means that when actin, which is another protein within cells, comes up to vinculin and it binds to it and pulls on the vinculin, then it'll open up, and then that is its activated state. And then it can bind to other proteins. In 2015, Horton and colleagues found this adhesome, which is just a database of all the interaction pathways between vinculin to other proteins. And here you can see the two main pathways are from vinculin to talin, and then vinculin to alpha-actinin. And our lab has been studying all these interaction pathways based on Horton's research, and knowing that vinculin is so important, we've already studied talin, and my project was to study alpha-actinin. So I wanted to learn what the effect of inhibiting alpha-actinin binding with vinculin has on the focal adhesions. And so normally alpha-actinin would come and bind, which is known as localization. It would just come and bind to vinculin when it's in its activated state. But if we inhibited it, we wanted to see what would happen to the forces acting on vinculin. So to do this, we did some research and we found that if you target the 133rd amino acid site on the Vinculin head domain, which I boxed in here, and you change it from an AAA, which is a lysine, to a GCA, which is an alanine, then it will be, alpha-actinin cannot bind to vinculin. So to do all this isn't really important, but these are all the steps that I went through. First, I just did a PCR to perform the point mutation, then I did a bacterial transformation, which just amplified all the plasmid DNA that I got. And then I did a mini prep to extract the DNA from the bacteria, and then these two steps to just confirm which, whether I got the right plasmids that I wanted with the point mutations, and then I sequenced them to confirm those findings. And then I would do a biotransfection, which is just taking the plasmid DNA that I got and putting them into mouse cells. And then I would pattern them, which is basically putting them and then shaping them like a star shape or a flower shape, as I showed before. And then some immunostaining to visualize the proteins within the cell. So our lab focuses on this Forster resonance energy transfer analysis, which is just FRET. The key takeaway here is that we have a tension sensor that we put inside of the vinculin protein. And it's just like this spring thing inside the protein. And if you have high FRET, when you analyze it, then that means low force. And then vice versa, if you have low FRET measurements, then you have a high force acting on the protein. That's just the main thing that you want to know. High equals low, and then low equals high. So my project is still continuing throughout the year, but something that we probably will get as a figure is this. This is from Dr. Rothenberg in our lab, and she was the one who did the analysis on talin, the other key protein that binds to vinculin, but she also studied actin, which is the one that pulls on vinculin. So she has 3 data columns. The first one is VinTS, which is just a control, and then A50I, which is when she inhibited actin, and then I997A, which is when she inhibited talin. And she shows images from the front analysis, and then the fluorescence is how you can measure FRET. And this is her quantified data as a box and whisker plot. So you can see that the control is what would normally happen, like the normal force distribution acting on the vinculin. And what happens is when you inhibit talin, then there's no significant difference. But she found that when you inhibited actin, then the FRET efficiency goes much higher, which means that, as I showed before, higher FRET efficiency means that there's lower force acting on the vinculin, which means that the key takeaway she found was that vinculin needs actin for forces to act on vinculin. And since vinculin is load dependent and only activates when load is acting on it, then that means that actin is necessary for vinculin function inside local adhesions. So my next steps are, I only got to DNA sequencing and I got the plasmid that I wanted, but then now I have to do the biotransfections and the rest of the steps to put my plasmid inside other cells and observe the effects that it has on proteins and measure the FRET. So my main goal that I wanted to learn from this was to work on lab procedures inside the lab, and I think I did that pretty well, learning all the PCRs up to the immunostaining. And I would like to acknowledge Carolina Livery, the Burroughs Welcome Fund, Chad Lloyd, my instructor here, and Arnold Tal, my mentor there, Dr. Brenton Hoffman for letting me into his lab, the NCSM Foundation for funding this, NCSSM for providing the opportunity, and the National Institute of Health R21 grant, which provided the funding for my project. And these are the works that I cited as images in my presentation. Any questions?
That's great. The proteins.
Yes, it's actually a filament.
A bunch of promines or something, or
yeah.